Okay, I think we can start. So it's my absolute pleasure to host today Professor Xavier Barril to give you some background on his CV. So he started biochemistry at the University of Barcelona, where he also did his bachelor's and his master's program, master's then in chemistry. The PhD was also at the University of Barcelona on the studies of protein receptor, ligand receptor interactions in the field of biochemistry. From there, he moved to Vernalis, a company that is well known in the field, located in Cambridge, UK, in the applications modeling team, and came back to University of Barcelona in 2005, where he was appointed research professor at the Faculty of Pharmacy. Besides that, he founded several companies. So the first one was Immunorix Therapeutics, then 10 years later, Oniria Therapeutics. And then he's also a part-time CSO in Gain Therapeutics, which recently also entered the NASDAQ. Needless to say that some patents are involved in all of those companies that Xavier holds. So we will see theoretical basic research applied to real-world applications. And I'm very much looking forward to your presentation, Xavier, on the robustness of protein-ligand complexes. Thanks for being here.
Thank you, Christoph, for the invite. Actually, it's a pleasure. I regret that I cannot be there in person, but I will be there soon, I hope. So, yeah.
I'll see you in April.
Yeah, and today I want to talk a bit more on the fundamental, applications for drug discovery, but not on any particular system, which is what we do in the company. It's more applied. So it's actually this concept of structural robustness and how it can be used in structure-based protein screening. So, yeah, that works. So everybody is actually in the structure-based drug discovery is actually after this parameter, right? This very simple parameter, the binding free energy. That gives you the dissociation constant, and if you could predict that, then you could design drugs in the computer. Actually, there's a bit more than that. You could also want to know about something a strange state called the transition state, and then if you have the relative free energy of those two states, the bound, the unbound, relative to the transition state, then you have everything. You have the kinetics, you have the thermodynamics, and you can really design anything. The problem is this is too difficult to calculate. I mean, it's lots of some of the people in the room actually are working on this, and we are seeing very positive developments, but it's a huge effort. And some years ago, we started thinking about what about something simpler, which is just this structural stability. And what it means, structural stability, is that potentially you could have two ligands that have exactly the same parameters in terms of binding free energy and the energy of the transition state, but they could have different landscape, the free energy landscape in the bound form. So one, for instance, could be very sort of shallow. So that means that the ligand could rotate and stay still bound, but have some ability to change conformation, orientation, things like that. Or you can have a ligand that actually has a very narrow minima, and it could only have very short vibrations and things like that. Macroscopically, this doesn't really matter. They could be the same, but it's an interesting question. And the thing is that it's a local property, so it should be trivial to calculate. And the difficulty in calculating binding free energy or to calculate the transition state is that these are very far apart, and going from A to B is quite difficult to do that in the computer. If you want to do it computationally to check if you're in a narrow minima or a broad minima, it's actually fairly simple. You start with the bound state and push it a little bit and check if you get resistance to displacement or not. And this is quite trivial to do. The question is if it's useful at all, right? And that's what I will be working on, talking about for this talk is first, how do we quantify structural robustness? The question is if protein ligand complexes are more like in this first picture, there's like sort of a shallow valley, or if they are more like in a cliff sort of situation, right? And we have also done some work on trying to understand what are the molecular determinants of that, but I will not be presenting that today for the interest of time. And then the most important question is if this matters at all for drug discovery. And so first, how do we quantify structural stability? This is something we do with dynamic undocking, or DUC for short. And it's an important first definition is what is the structural stability? Because if you imagine this ligand, most of it can kind of stay bound and be fairly robust, but other parts may be rotating or moving, right? So I know from social media that if you want to be popular, you have to use cats in your, so that's what I'm going to do now. The question that, this kitten is really wondering if this roly-poly toy is structurally stable or not, because part of it is moving and part of it is static. So, and we know that that protein ligand complexes, part of they are in part mobile, right? So we know that from crystallography. In some cases, you even have the same molecule binding in different orientations. More frequently, what you have is a part of the molecule can adopt different orientations. And actually, when analyzing the electron density, it's fairly common to be able to fit the molecule with multiple occupancies. And in this very interesting paper, they quantified this like 1/3 of the structures of the crystals actually have unmodeled electron density that could correspond to some degree of disorder. So, but that's not what we are, how we define structural stability. We want to see if the protein has some part of the protein ligand complex has some interactions that are really stable, right? So that we know that there will be some mobility elsewhere, but at least there should be an anchoring point. And for that, hydrogen bonds were the obvious place to start, because obviously they have like a very well-defined minima that in a matter of almost 1 angstrom, you completely lose all the energy that you can gain by forming the hydrogen bond. So it's unlike van der Waals and hydrophobic interactions that are more permissive. Here you have a clear minima. And some years ago, we identified that depending on the local environment, you can actually create a trap where the hydrogen bond, if you want to break the hydrogen bond, then the waters cannot come in, and that creates a transition state. And this can actually be of very high magnitude, and it actually even impacts on the kinetics of the ligand. So that was actually why we started investigating this. Obviously, most protein ligand complexes do have hydrogen bonds. Hydrogen bonds are almost universal. And even the smallest ligands, like in fragment, and we also have a keen interest on fragment-based drug design, then they also form hydrogen bonds. And this is something that Curie quantified here. So then what we will be doing is to actually look at hydrogen bonds and whether they are robust or not, okay? And this is from the computational side is very convenient because you can do very short simulations. You just have to, you have an ideal hydrogen bond, you pull 2 angstroms, and it's completely broken. So you don't need to more than that, just 2 angstroms. And this is also a trivial reaction coordinate. When you try to use any form of bias in molecular dynamics, you have to think very carefully how you're going to associate your ligand or dissociate, how you're going to tell the simulation to actually proceed through a certain pathway. Here, you're just breaking a hydrogen bond. So a simple distance works, and it's something that you can automate completely. And also, it's a relatively simple energy landscape because it's such a short distance. You can only change like the orientation, right? You're increasing the distance, and it can go a little bit in this direction or this other direction. But in reality, it's not such a complex process. And actually, because you're interested in a very local property, you don't need the whole system. You can get rid of most of the protein and just keep the surroundings around this hydrogen bond to make sure that you capture this local effect, but you don't care what happens, I don't know, 15 angstroms away, right? So with these considerations, we created something that we call dynamic undocking, and it's just a form of steric molecular dynamics, but we'll give it a name because the way we use it is a bit peculiar. Okay, so what we do is first we take our protein of interest, and then we define an interaction on the protein side that we think is important. So in this case, it's this aspartate on HSP90. And this could be our ligand. And we start with a distance of 2.45 angstroms. And then we run molecular dynamic simulations with steering these 2 atoms to form the hydrogen bond. And we measure the force that we have to apply to separate those 2 atoms. And because we're doing this at constant velocity, we can also get the work. And essentially, you run this simulation and you get this work profile. And this is telling you that the ideal hydrogen bond distance is 3 angstroms in this case. And to reach 5 angstroms, so just these 2 angstroms, you actually have to put in 8 kcal per mole, which is a huge amount of energy, actually. And that's a property that we've been looking at. So the cost, the amount of energy that you have to put into to actually break a hydrogen bond in going from 3 angstroms to 5 angstroms. Why we didn't call it steric molecular dynamics and so on? Just because first we use a part of the protein. We don't use the whole protein. And secondly is because we do not try to get free energies. Okay, it's an important distinction. If you want to get free energy from steric molecular dynamics, you should run, as close to infinite simulations as possible. You could get this sort of dispersion, and then you could use the Jarzynski equation to convert this into a binding free energy, which is essentially the Boltzmann average of the work. It would be the free energy. Okay. In ideal situations, you can do that, but we don't want to run to completion first. So we want to run 100 steric molecular dynamics. And more importantly, also when you run steric molecular dynamics, frequently you have something like this, where you have 98 simulations that you look like fairly similar to this one. And then you have these 2 simulations that are giving you a different profile. And this, it's difficult to tell why it happens. It could be, for instance, just to give an example, you're pulling and it's very difficult, but in this simulation, for whatever reason, when you were at half pulling this, then the ligand rotates a little bit, and then breaking the hydrogen bond becomes now much easier, right? This rotation, which is what the ligand would do in a natural situation, is not included in your reaction coordinate, which is just pulling the atom. And then it's very rare that you see that, okay? So when this happens, it's telling you that the reaction coordinate is incorrect, and this you can no longer use that because you should repeat the simulation using a different reaction coordinate, essentially. So we don't want to do that. So what we are going to do is just to take the lowest value of all the maxima of each for each trajectory. We look at the highest point, and then we take the lowest in all the simulations. So in this case, this could be the value, about 4 kcal per mole. In this case, it would be here, which is, I cannot even read the number, but it doesn't matter. You get the idea. Is that we do not try to get the binding free, the free energy of that process. We're trying to get an approximation to that, which is a work value. And that's it. Okay, so because we want to also be able to run only a handful of simulations and decide if this is easy or difficult to break. Okay. Right. So this is was more technical, but now we can actually come to the interesting part. Is then our protein ligand complexes really structurally stable. And this is a work done by Maciej Majewski, a very talented PhD student in my lab. And he took a whole set of protein ligand complexes, and for each protein ligand complex identified all the hydrogen bonds, and then performed these simulations that you've seen for each one of them, right, to completion. So lots of simulations. And then out of 345 hydrogen bonds in about 150 complexes, this is the distribution you get. A third of them are less than 4 kcal per mole. They can break fairly easily. But actually, 57% actually have a value that is higher than 6. And you can see that in some cases you go to very, very high values, greater than, larger than 15 kcal per mole, for instance. These are extremely robust hydrogen bonds. They are very difficult to take. They would have a very long half-life, actually, right? So yeah, this was done with the iridium data set. But if you then, instead of looking at each individual hydrogen bond, you look at individual ligands, what you find is that 75% of the ligands have at least one hydrogen bond that has more than 6 kcal per mole of work QB. So it's actually very, very common that ligands form robust structures with a protein. Right. So, and what is interesting is that this is not depending on the size of the ligand. So if you take fragments that are on average like 200, 250 molecular weight, you get, this is a smaller data set, so only 124 hydrogen bonds in 26 complexes from the Seraphic data set. Actually, you get essentially the same statistics. And actually also sometimes extremely, extremely robust hydrogen bonds. Yeah, so that means that even small ligands can form these very, very static structures. Is it necessary? No, it doesn't have to be like that. There are very potent ligands like this one. These are two allosteric inhibitors of the HIV reverse transcriptase, and they are 6 nanomolar and actually 0.6 nanomolar, so extremely potent. And they have a single hydrogen bond, and the hydrogen bond is very labile. Into 3 kcal per mole is all you need to break. So actually, these complexes are probably fairly dynamic. When they are in the protein, they probably can break the hydrogen bond, move a little bit, rotate, come back in, and so on. So there's no reason, as I said, why the complex should be always stable. And actually, from an entropic perspective, this should even be favorable to have a sort of disordered bound state. But in the majority of cases, what you have is at least one robust interaction that makes that anchors the ligand. Okay. So then we can use this property in different things. And the thing that we have used more, the first thing that we tried and what we are working more on is virtual screening. But you'll see that it can be used for also different things. There's something that was published a few years ago, and Sergio Ruiz was the person doing it, and now in Australia. Again, also I've been blessed with very good PhD students, so that was a very good piece of work. So these initial investigations are on 3 different systems: a kinase, a GPCR, and the protease, and using the work necessary to break a hydrogen bond to distinguish between true ligands and decoys. And actually you get very good enrichment. These are the typical ROC curves where you see that, if you're above the straight line, the diagonal, then you're getting enrichment. And we're getting good results in all cases, clearly comparable to doing docking. But the thing is that you don't have to do that instead of docking. You will have to do docking anyway to actually generate an initial binding mode. So and with docking, or then if you use other post-docking methods like MMGPSA or MPVSA, what you're doing is you're taking into consideration the whole set of interactions between the protein and the ligand, and you're looking at the minima, right? At the equilibrium situation, at the best docking pose. Whereas with dynamic undocking, you first need this binding mode, and then you're only looking at a single hydrogen bond. In this particular system, on this work, we only look at one hydrogen bond, which was always with a predefined amino acid in the protein. And you're looking at the cost of breaking the interaction. So you're looking at something out of equilibrium. And so to show that they are really complementary is what we have in this graph, where you have each point corresponds to one protein ligand complex, and then on the x-axis you have the work QB. So the higher the number, the more difficult to break. So allegedly it should be the better. And in the y-axis, you have the docking score. Lower numbers, the more negative numbers mean better score. So the down here and is better, and then down to the right is the best position. So when you have this, means you have a good docking score and also difficult to break. And here the enrichment factor that you get is much better than on average. So they are complementary views, right? So one is looking at the whole set of interactions. The other is telling you this forms a robust interaction. If you have both, then you're in better chance of finding a compound that is active. Okay, the active could be the red dots. Okay. So doing that, initially we did a fragment screening, actually looking at 280,000 fragments, did docking first, and we took the top 500 top scoring, filtered on diversity and solubility, and then out for each one of those we ran all the simulations and classified this into 3 categories: the strong binders, more than 6 kcal per mole, the weak binders, less than 3, and medium. And then we bulked as many compounds as we could from each one of the categories. Out of this set, 21 we could beat by, and 8 were active. So that's a 40% hit rate in fragments. The random hit rate for fragment screening in that particular protein with the same setup was 4%, so that's a tenfold improvement over random. But also interesting, we bulked some other compounds in these categories, and only one of them was active, and it actually was like almost strong, right? 5.6, the threshold was at 6. So it means that if your compound doesn't form a strong hydrogen bond, at least in this system, then it's very unlikely that it will be active. And actually a large proportion of the molecules that come out of docking form very weak hydrogen bonds. So this is a good property to use in virtual screening. this was done in collaboration with Vernalis. They also got us some crystal structures, and it was very nice to see how that it could work. But since then we have been using that in every virtual screening campaign that we do. And now I want to talk about what we are doing today in terms of virtual screening. For us, the new challenge is to actually be able to change the scale of virtual screening. We usually would screen about 8 million compounds that you can buy at Molport, but as you know, now with this real space or on-demand chemical collections, the space has grown to 41,000 million compounds, or if you speak American, it's 41 billion, which is always simpler. And here we investigated how to access that, right? And yeah, I want to stop a moment just to say that the company that actually made this possible is Enamin. And as you know, they are based in Kiev, so they had to stop, all activities due to the invasion. So I hope they do well, because they've been doing an amazing work. When they set up these collections, nobody could believe that you could actually buy these compounds, because they say, okay, yeah, you say you can do them, you say you can synthesize them, but in reality, then you will try to do the synthesis and you will fail, and it will take you a long time. And it's not like that. So actually, we bought a lot of compounds from them, a very nice collaboration with them. More than 90% of the compounds that we ordered, they delivered, and in 1 month. And actually for a price that is about the same as if the compound is in stock. So that is really transformative for virtual screening, because virtual screening, we have always been like the, like the Cinderella of if you're a company, you do high throughput screening. If you're a poor academic lab, then you do virtual screening. The pharma companies have like 5 million compounds, actually physically, they can screen. So that was about the same range that we could do before. Now, if you do virtual screening, you can do 10,000 more than they do. And this is actually growing. Their expectation is that in 2025, they could have like 1 trillion, so 10 to the 12. So for each molecule that the company can have in store, we could have 1 million compounds, right? So 1 million times more doing virtual screening than doing physical screening. So we have to seize that opportunity, but to do that, we have to change how we're doing virtual screening, and that's what we're trying to do. Unfortunately for us, we're not the only ones that are thinking that direction, and some already had similar ideas. But what is clear is if you look at how the chemical space grows, right? It grows more than exponentially with the number of atoms. So the whole drug-like chemical space, molecules with nitrogens, oxygens, and carbon, and so on, if you consider up to 4 atoms, it's only 100 molecules. If you go to 7 atoms, it's about 10,000. When you reach this level of fragments, which is 8 to 12 heavy atoms, you already have 145 millions, but that's manageable. The problem is when you go to the drug-like space, it's 3 times to the 25 or whatever. This is based on this GDE. It's just huge. We cannot do all of this, right? And nowadays we have this 32 billion, 3 times to the 10. Doing the docking of all these compounds would be impractical, and actually they are not even enumerated. So you cannot do that. But if you stay in that space, there's about 4 million compounds that of this size. So this is completely doable. And even if that grows, there's this limit of 150 million compounds on that space. So this is where we propose we should stay. Start at the bottom, do the virtual screening on fragments, and then when you find something interesting, you do a search for molecules that contain parts of this fragment. And also that was not possible until recently, and thanks to Matthias Rari group, now they have developed this SpaceMax, which is fantastic. It does a substructural search on these huge chemical spaces and retrieves all the molecules very quickly. So we use that in collaboration with them. And then also we say that we shouldn't do just docking, we should do a hierarchy of methods, right? So how to get better prediction. So, and this work is actually, we have to credit Serena and Moira, two former PhD students in my lab, because it's not the same, but it's heavily based on a paper we just published on the evolution of and navigation of chemical, fragment chemical space. So what did we do? We took these 4.1 million fragments that you could theoretically buy, and we did docking first. We optimized the conditions to maximize the enrichment factor. This is on BRT4, which is a well-known system, and we could only get like almost 5-fold enrichment factors, so 5 times better than random. So we end up with 362,000 fragments out of docking, and we cannot work with this number. So we had to do clustering. I think one thing in the future, it is something that I discussed with Thierry and also with Johannes. It's about the possibility of using other filters, right? But essentially now what we had to do is to just do clustering, and we go from this huge number to only 2,000, and then we use MMGVSA. It's difficult to get the good enrichment with MMGVSA because the active here and the rest of the molecules actually have a very similar profile. You can only cut this tail essentially. And then we use dynamic undocking. And with dynamic undocking, we can actually reduce a lot the number of candidates, even though they have good docking score, they have good MMGVSA score. Actually, we keep only the molecules that have a work QB greater than 8 or 7 in that case, and that's only 4% of the candidates. So that really limits the number of molecules we have to consider. And this is still ongoing, but essentially we did that. We did this virtual fragment hits, and those were then we perform a substructural search on the biggest space. So for instance, we get 17 million molecules that actually contain this particular substructure. And then we do, again, the docking, clustering, MMGVSA, dynamic undocking, and we end up, in this case, buying 5 compounds. And then we experimentally test them. So that would be like the worst situation. We only get 1 active, 20%. For comparison, we did the same thing with real fragments that are described as active in the literature with crystal poses, and with the scaffold of existing drugs, 3 compounds that are in the clinic against this target. And actually, what is really encouraging and this is, as I said, we're still working on this, and we'll have to see how potent they are and so on. But at 1 micromolar, which is not a high concentration, in delta TM, we have more than 1 degree. And we get 60% almost from this set, which is very similar to what you get if you look at the scaffolds of existing fragments or actually drugs. So we think that this is definitely a viable strategy. You can do completely virtual. You don't need any experimental data there. And we think that the dynamic undocking that allow us to actually reduce a lot and focus on the ones that have more robust interactions it has to be used to actually have this level of success. And this is work done by everyone in my lab. So that was really a completely collaborative effort. It was a really very nice experience from my side at least, but it was mostly led by Alvaro and Marina, two PhD students in my lab. So we'll try to finish that and get better additional data, binding data, and also crystallography data. But it looks like very, very encouraging for the future how to handle these huge chemical spaces. To So actually, and then in the last sort of 10 minutes, I'll try to show two other applications of dynamic undocking and also explain why we think it works. So the other one is binding mode, and this also was done by Mathieu. And essentially, what we saw is that if you have several candidate binding modes, and then you rank them in terms of how robust they are. So if they form a very robust anchoring interaction, then actually you can use dynamic undocking to identify the true binding mode, distinguish it from the rest. So I'm not going to go into details, but that would be the result of docking for fragments. And this is the result of dynamic undocking for fragments at a different cutoff, deciding that the correct pose is at 1.5 angstroms here or 2 angstroms. So we get, like, depending on that cutoff, 50% or 70% of the fragments correctly predicted. And this is, again, just based on the prediction of if one part of the molecule is forming robust interactions. You're not looking at the rest of the molecule. So when you look at bigger molecules that are not fragments, the performance is similar. It's about 75%, but then docking wins or gets almost the same performance because it's looking at the whole interactions, right? So where do we fail? Often what we fail to predict is the part of the molecule that is not forming the anchoring interaction. I mean, let's for instance, in this case, this is the robust interaction and we get it right, but the other part of the molecule, dynamic undocking, knows nothing about that. It's not even, it's not accounted for, and then we get it wrong. So again, using docking, taking several binding modes and then just removing the ones that are not forming very robust interactions is a good way of improving binding mode prediction. And now we come to the holy grail, which is binding free energies, right? And this is again done by Alvaro in our lab. So can this be used to predict binding free energies? No way, okay? So we know that from the beginning. If you take binding free energies from a diverse collection of molecules on different targets, and you measure the work to break these interactions, and you plot it against the binding free energy, you get a scatter plot, so no correlation whatsoever. But what about a change in binding free energy, right? So in lead optimization, we would be happy to just say these are two compounds that are very similar, and one is a lot more potent than the other. So can we predict that at least? So these are what are known as activity cliffs. So Alvaro took several sets of activity cliff data sets and then computed the work necessary to break these interactions for each one of those compounds, and then was looking at if we could predict the change in binding free energy. And to make a very long story very short, this is the Matthews correlation coefficient. Just tell us if we can rank the compounds properly, and then we should look at this Jasinski. So we looked at different ways of doing it. We also compared with MMGBSA and with docking score, and it was like a very strange feeling, right? Because you get 3 data sets. So this is a series of compounds. These are all, remember, these are all similar molecules, right? So we have for CDK2, the kinase, we actually had 4 different series of compounds. And on this series, we get like very good performance. And then on base and HSP90, also we're getting very good performance. But then in other cases, we get no predictive capacity. And that was really intriguing, right? So even though you could say, of course, you're better than MGBSA in all cases, you're better than docking in all cases. For us, what was really interesting is why this works and why this doesn't work. And so, in other words, what is the scope of dynamic undocking for prediction of relative binding free energies. And I don't have time to show everything, but essentially that is the main message. What we did, we look at the whole dissociation process using random accelerated molecular dynamics. And what happens is there are some complexes that you have intermediate states. So you have a bound state, and then you have an intermediate state, and then you have the dissociation. And in these situations, dynamic undocking cannot work, right? Because we're only looking at this first barrier, right? So it may be very steep, but if you're then falling back into another binding mode, you just run this, you don't know what's going to happen next. So when you have a family of compounds that they have intermediate states, dynamic undocking doesn't have predictive power. But when you have this sort of situation where you have like a two-state model, right? It's either bound or unbound. It just either has a large barrier and then it's off. Then you can actually predict the two compounds. The one that has a steeper potential is actually because it has a deeper minima, and that's why we have this predictive capacity. So actually, especially now that there are so large collections of compounds, there's nothing wrong with this sort of complexes. They are perfectly fine, like the allosteric HIV reverse transcriptase inhibitors. They have this sort of profile for sure, and they are fine drugs, okay? But if you are in the situation where you have a complex or a series of compounds that actually bind with this profile, then you don't need to actually look at the whole energy landscape. You just have to look at this very narrow space here. So what we would like to propose is that you can use dynamic undocking for virtual screening anyway. Even in this situation, that works. But if you want to use it for binding free energy, it has to be like that. So if you're doing virtual screening and you're actually selecting for something, then selecting for molecules that have this profile is a good thing, because it should help you later in the optimization process as well. Right? So that's what we're thinking nowadays, that you could sort of do robustness-based molecular design rather than binding free energy-based molecular design. So coming back to the questions, how do we quantify structural robustness with dynamic undocking? Is a good way of doing it. Are protein-ligand complexes robust? Mostly they are. So this is actually a lot more frequent than this other situation. And yes, and the properties is actually useful for binding mode. Definitely for virtual screening, it removes 80–90% of the docking false positives. It's very well tested. It can also be used for binding mode prediction and for binding relative binding free energies in some cases, and actually also for kinetics, although we cannot. There's not a lot of data to check, but obviously it should work better for kinetics than for actually for binding free energies. And with that, I'm coming to the end, and I want to acknowledge everybody in my current group and in my past group, and our close collaboration with, especially with Carlos Galdeano, who's actually not in this picture for some reason, but also Jordi Juarez and people at Vernales and the different companies that I also collaborate, and everyone for your attention. Thank you very much.
Thanks a lot for the presentation, Javier. Very exciting. I just take the liberty and ask the first question, because you just finished with the intermediate states, and you also mentioned the reverse transcriptase of HIV. Naturally, I'm very interested in allosteric proteins. So can you comment a little bit more on how one could use that to maybe even find allosteric sites or probe them with blind docking? like cryptic sites by extending maybe the MD run. I don't know.
Yeah. What we have been targeting allosteric sites for many years, and we always do first these molecular dynamics with mixed solvents that allow us to identify binding hotspots. And what is really distinctive about this method, the way we use it at least, is that it tells you if there's a polar binding hotspot. Okay, because hydrophobic binding hotspots are trivial to see. You have a hydrophobic surface, you're going to have a nice interaction there. Hydrogen bonds, on the other hand, I mean, the protein is completely covered by hydrogen bond donors and acceptors, but there are not so many hotspots for hydrogen bonds. So for us, a good binding site, allosteric or not, it combines some hydrophobicity with some key polar interactions. And when you see that you have in a small space, you have hydrophobic hotspots and polar hotspots, then that's what for us is an indication of a good druggable binding site. And then when we do virtual screening, we first impose pharmacophoric restraint Undocking always. And the pharmacophoric restraints that are really useful are the ones that look at hydrogen bonds, because again, the hydrophobic hotspots are broader, so they are not so, they do not impose a strong restraint. But the polar hydrogen bonds, they do impose a strong restraint, and that helps a lot. And after that, we do dynamic undocking on the same hydrogen bond. So this property, remember, is a property of the protein-ligand complex. So it's not a property of the surface of the protein or a particular atom on the protein. Depending on what comes to form interaction with an atom in the protein site, you can have a robust or a very labile hydrogen bond. Okay, so we use it at the virtual screening stage, not in the druggability, in the identification of druggable allosteric sites. Okay. I hope that answers the question.
Yeah, thanks a lot. Other questions? Please raise your hand or just open your microphone.
There's one in the chat.
Yeah.
Oh, yeah.
What docking function you are using, and whether you're using any QM, I guess, approach somewhere in your methodology. Is that QM or QZM?
Yes, it's QM.
Okay. No, I wish we could use QM, but no, I mean, you see the size of the libraries we're looking at. Traditionally we were using only 8 million, and now for each, you have 4 million fragments, and then for each fragment that you identify as good, then you go search in that big chemical space and you retrieve 10 million, 20 million molecules. So unfortunately, not. I think now machine learning with QM, that maybe at some point you can get QM quality predictions with machine learning, but no, at the moment we haven't. And to be honest, something that has to the robustness of the hydrogen bond, there may be some things that we are not accounting for, like polarization, right? Of the hydrogen bond can be polarized and so on. But the bulk of it, I think, is essentially comes from whether water molecules can insert in between the hydrogen bond or not, and
Yes.
also on what happens when you bring this hydrogen bond, if it's facing something else or things like that. So I think QM obviously would improve the quality, but the most of the effect you don't need. It seems that like you don't actually need this QM calculations for that.
Okay. And what which docking functions are you actually using for generating the poses or the
Okay. For Yeah, I'm attached to a particular docking software. It's called ALLODD because it was developed at Vernalis when I was working there. I find it works very well, but not because the scoring function is particularly good, but because it allows us to do this template tether docking and allows us to do pharmacophore restraint docking. So the scoring function is not, I wouldn't say is particularly good. It's probably just as bad as the majority of docking programs. But
Yes.
and that's why we always supplement the docking protocol with empirical information to make sure that we tell the program, you must fulfill this hydrogen bond, you must place something in that particular subcavity, things like that, and that seems to sort of alleviate the problem of the scoring function, which is bad as any other docking.
Thank you for your answers.
Thank you.
There was another question in the chat from Thierry. How long do these runs take? Minutes, hours, weeks? Months?
No, actually I have, let me see if I can, I've stopped sharing now, huh? Apparently.
Yeah.
Okay, so I think I have the answer. Here. I'll try to share again. This, sure, okay. So this is the answer to your question. We run very short molecular dynamics. We first have to run molecular dynamics to equilibrate the system, and then we do stirred molecular dynamics, which are only half nanosecond length. So for each calculation, if you take this is more like a Tesla or P1000, so this is a good GPU. It takes each stirred molecular dynamic, it takes only 6 minutes. Of course, you have to run many to actually get the confidence. If you want to actually get binding free energy, you should run at least 100 to 200 of them. And in between, you would run some molecular dynamics to change the starting position and so on. But if you're doing virtual screening, what we're doing is we do molecular dynamics, one stirred molecular dynamics, and if the value that we get is already below the threshold, we stop. So in most cases, in 10 minutes, we have finished for virtual screening. If you want to then actually get quantitative predictions or proper, like try to get the binding free energy or the free energy of those transitions, and then you should run many, and it would take hours. But this is like really doable. So that's why for each one of these virtual screening exercise, I think we did 400 molecules, but we would love to actually be able to do it on a much bigger scale. And one thing that we really will be looking into is how we can use all the simulations we already have to use machine learning and then predict for new molecules what would be the outcome of that. So we would save a lot of time, and then probably we could expand to thousands and thousands of cases.
Okay, thank you.
Thank you.
So Johannes, you also had a question.
Okay, what is your view?
I can just say it. So I was just wondering, in your experience of the enamine real space, I mean, it's a huge chemical space. I was just wondering how do you see like the beauty of this chemical space? So is there many compounds that you say these are really nice, or is there a lot of?
Yes. So yeah, it's a good question. Actually, one thing we have introduced is a filter on the number of rotatable bonds, because we found that a substantial proportion of them, I don't know, 20-30% of them, had like very flexible linkers in between rings. So we have a limit because docking them, first it takes more time. Secondly, they are not really interesting. But actually, no, they apart from that, for the rest are really, really nice. I mean, what they do is they do either only one reaction, merging two building blocks, or two reactions in one pot. That's what they do at the moment. I presume that their plans were to expand that. But they are fairly small. Majority of them actually do not, they are 300 something to 400 and so on. So they still have a space to actually make molecules bigger, and their properties they look good. I mean, but this is just my visual assessment. Okay, and as I said, it could be ideal if we could actually reduce these numbers by looking at properties like solubility, like I don't know, toxicity predictions, things like that, and just get rid of them before even we do docking. That would be ideal. But they look very, very medchem friendly.
Okay, thanks.
Thanks. Other questions?
Actually, I have a question. Can you hear me?
Yes.
That's hopefully not the question.
No. I was thinking, how good does this work without having any idea about binding site on a protein?
Yeah. So that's so this is something I didn't present today, but we when I started my group at the university, I wanted to work on a bit exotic binding sites, let's say. Not I didn't want to work on kinases or GPCRs and so on, where the companies have a lot of knowledge. And actually, very quickly, I realized that we needed proper methods for drivability prediction. And this is something that we did some years ago. We used this method called MD-mix, molecular dynamic simulations with mixed solvents to identify binding hotspots. And then a binding site is a collection of binding hotspots for us. And we have used that in many different projects. We have also validated the method retrospectively and so on. So we are fairly confident about this method. And then when you have that, then you start with the virtual screening. But yeah, if you don't choose your cavity wisely, then obviously there's nothing. You shouldn't expect any result.
Thanks.
And I have another question. Of course. What do you think? How good would this work for non-drug-like molecules like carbohydrates?
That's yeah. I thank you for the question. Actually, we tried when Mathieu did his study, we tried to also look at the behavior of different types of proteins. Let's see if I can find that. Yeah, here it should be here. Yes, and we had a set of 14 carbohydrate binding proteins, which in total made 95 hydrogen bonds, because obviously there were lots of the carbohydrates have hydroxyls, no? And they make lots of hydrogen bonds. So what we found here is that you actually can have very robust hydrogen bonds in some cases, but it looks like on average maybe the ligands are less, a bit less. There's a little bit smaller fraction of ligands that form very tight contacts on one particular area. This, if you compare it with enzymes, which will be 82%, here you have 64%. But I'm not sure if, with only 14 complexes, I'm not sure if that statistically is meaningful. You see that this one thing that is interesting is we are not looking at water-mediated hydrogen bonds, because the technique, the way we have implemented it, is still not able to do that. So we're only looking at direct interactions. So I wouldn't be surprised if actually some of these are water-mediated hydrogen bonds that are actually quite robust, or other form of interactions. But many of the hydrogen bonds, so obviously these proteins form lots of hydrogen bonds, a lot more than the average. So they also have a higher proportion of labile hydrogen bonds, right? So 40% about are very labile.
Okay, maybe we can discuss this at a different time point, but it's very interesting here to differentiate carbohydrate binding proteins that are from mammalian or bacterial origin, because usually, so there's a different underlying mode of co-evolution, which either draw them into weak recognition or into very strong recognition. Yeah, but we can discuss this at a different point. Are there more questions? Yeah, Julian.
Yes, thank you very much for a beautiful talk. I was just now going back to the problem when you sample the binding site with a small well, and you have a different ligand with intermediate states where you need to actually sample many more wells to get free energy. I was wondering if you use coarse-grained MD, for example, or if you lower the resolution of your MD, if that would actually help to sample those other states, or if that would be actually a way to go. In other words, is this now, did you reach the limit of MD with this type of application by pulling a little bit out the ligand, or can you still tweak around it?
No, there are methods that can actually give you the binding free energy for a complex of any anesthetic landscape. But the problem then is that you have to sample the whole thing, right? Frankly, we don't want to do that. With dynamic undocking, you cannot do it. With dynamic undocking is only meant to look at this very short distance. So what we propose actually is that you should run something. You don't even need to calculate the binding free energy. You can run things that are fairly rapid, like random accelerated MD, and identify if there are transition states involved. Intermediate states involved in the dissociation. If that's the case, then dynamic undocking is not the method for you. And then don't use that method, use something else. That will be a lot more expensive, and but also a lot more rigorous, right? And then you can tackle this problem differently. But if you are with a system that has this property of being just sort of two-state, then what we're saying is you don't need to go to very expensive systems, very expensive simulations. You can do this much faster and cheaper simulations and already get good signal. Okay.
I see. Thanks. Yeah. But do you think like there's other sampling methods or other strategy for the MD would not really help you in that particular problem?
Yeah. I mean, for instance, Francisco Gervasio I mean, binding free energy prediction of complexes with metadynamics it works. I mean, but it's very complicated because you have to find the reaction coordinate, and you can do it with a limited number of compounds. At least, as far as I know today. Probably that's going to change in the future. It's going to improve. So it's not a hopeless problem at all. I mean, it's something that can be done, but it's a different scale of complexity. That's what I'm saying. That for certain cases you have. Something that you could think is very difficult, which is predicting the relative binding free energy, it actually becomes a local problem. It's just seeing if you have a steeper wall or not so steep. And that is something that hasn't been recognized before. So that's what the message, right? That sometimes you can use this method. And you could even, maybe you'll be in the lucky situation where you have a series of compounds that have an intermediate state, another series of compounds that do not have an intermediate state, then stay with the second one and use the calculations that we're doing, because it will be much simpler to get the predictions right. Yeah, but we're not solving all the problem at all, right? It's only a fraction.
Any other questions? Yeah, please.
Hello?
So yeah, please go ahead. So who Bruno?
Can you hear me?
Yes.
Yes, we can hear you now, Bruno.
Hi, nice to see you again, Dr. Xavier. Hello, everybody. Thank you for the nice presentation. I only have a question. I was giving a quick look about this paper about the undocking, and I were really well, I figured out most of the things you talked about, but I want to ask you about how have you evaluated not only the hydrogen bonds in this dynamic undocking? I imagine that the necessary energy to disrupt a salt bridge is higher than the hydrogen bonds, but I don't know, not only salt bridges, but maybe stacking or van der Waals interactions. Have you ever thought about it?
Yeah, it's a good question. So first, I should have said that when we talk about hydrogen bonds, there are hydrogen bonds that are charge reinforced and hydrogen bonds that are not. So we have all sorts here, and we actually separated them, and we looked at neutral hydrogen bonds that were mixed. So one was positive, the other was negative, or sorry, positive, the other was neutral, or the other way around, and also at salt bridges. And actually, there's no difference in the distribution. So again, this may seem counterintuitive, but actually, what we're looking is at the barrier of this transition state between the one that is formed and the one that is separated. And this has to do a lot with the local environment, if waters can come in, what happens with the other, with these hydrogen bonds as they move apart and so on. Actually, for hydrogen bonds, on average, it doesn't matter if you have a charge or not. If it's that's something we looked with Machi. And then as for other types of interactions, of course, there are many more interactions. One thing we are not doing, for instance, and we should probably look at is CH hydrogen bonds. That would be interesting. The other types like stacking and more van der Waals interactions, they actually, by definition, almost they are like easy to displace. And then if you want to get this value, you would have to look at longer distances, and you wouldn't have such sharp profiles. So I know that there's a paper now somewhere looking at structural stability in the context of protein robustness. And they actually, what they find is also that hydrogen bonds are the main contributors to robustness. So this is based on this, what's his name, is where the single molecule pooling experiments. And one thing is how difficult it is to unfold the protein. And this has to do mostly with the hydrogen bonds, not so much with the hydrophobic interaction. The hydrophobic interactions contribute the majority of the binding of the free energy of the process. But actually, whether you have a sharp profile that is difficult to break or a more shallow profile that you don't have to apply a lot of force, that's determined by the hydrogen bonds. So again, we have to make a distinction between what makes a contribution to the binding free energy and what makes a contribution to robustness. And hydrogen bonds are clearly the most important ones for robustness, not necessarily for the binding free energy. So that may seem a bit counterintuitive, but
okay, thank you so much, Doctor.
Yes. So
thanks. More questions for Javier?
Yeah, maybe I have a question. You mentioned a few times that you were interested in incorporating machine learning in this as well at some point. Mm-hmm. I was wondering if you could maybe elaborate a little bit more on that. Were you sort of envisioning training a machine learning model based on a lot of examples of these undocking runs so that for a future compound, you wouldn't even have to undock it anymore, but you could just predict it based on the machine learning model, or is it more some sort of hybrid?
That's exactly the point. So there have been already a number of papers where people do docking, and then they train a machine learning model to predict the docking score. And it makes sense when you want to go for these huge collections, right? Because docking is fast. But when you're talking about, 1 trillion molecules, but docking, then essentially and that works very well. So it gives you an improvement. I can't remember, but it's in the order of 40 to 50-fold improvement in performance, because you only have to dock a subset, then you train, and probably you can do that in several iterations. And then you can dock only a subset of the total and get good predictions for the vast majority of the collection. Dynamic undocking is a lot more expensive compared to docking, and it would be ideal if we could like have a good method that, you know, given the complex that comes out of docking, and a definition of this is the hydrogen bond that I'm going to pull. What is the work profile that you're going to get? Because we have thousands of examples of that, right? So we can. It's not a problem for us to run, I don't know, 500 complexes, but it becomes a problem beyond that. So it would be nice to actually combine those two things and then get that performance. That's the idea.
Right. And is this something that you've already tried before? And
yes, we had Torben, a student from Germany, coming from for a while, and he started with that, and it was actually it was our first experience in machine learning. So I loved that, and it was really interesting. And he was doing lots of interesting different implementations and so on. The thing is, and that's what I fear the most, is that we don't know when we are outside the scope of the model, and when
that was going to be the next thing I wanted to ask you.
And so when we implement something, we want to make sure that we have a good way of telling, okay, that's a prediction, and that's the confidence in the prediction. And it's very simple, because if the confidence in the prediction is bad, then we can do the calculation, right? But we need to have both things. Otherwise, it's not worth it. No.
Yeah. Okay. Cool. Thanks.
Thank you.
More questions? I think also in the interest of time, we close the session here. Thanks everybody for participating, and thank you again, Javier, for this wonderful presentation. I enjoyed this really a lot.
Thank you very much.
Thanks.
Thanks.
Thank you. Bye.
Bye-bye.
Thank you. Bye-bye.
Bye. Thank you.
Bye-bye. Thank you.
Bye.